All, this is Dr. Mubin Sayed from Drbeen.com. Welcome to one more show. The discussion today is about the spike protein on exosomes. Exosome are little particles on exosome, and then this exosome with spike proteins in the blood of the persons who are vaccinated with Pfizer BioNTech vaccine. And this may be the case for Moderna as well. Now, those who feel that the spike protein may be harmful or hurtful to some tissues, they may be concerned with this. The discussion from the authors is that this is the mechanism by which the messenger RNA vaccines produce antigenicity or immunogenicity, not antigenicity, immunogenicity or immune response. So let's look at the mechanism that they have described in their study. So here is the study. Before this is Drbeen.com. If you would like to have access to about 1,000 videos, which are premium content, there is a link in the description, which is almost free. It is so low in price. Okay, with that, this is the study. The study was actually received on July 1, 2021. It was accepted September 15, 2021. It was published November 8, 2021. I thought this study should have gained a lot more traction because it shows something that has been a point of research for a long time and even point of theories. And that is, could spike proteins be present in the blood of the vaccinated individuals? And this shows yes. Number one. Number two, it shows that those exosome and the spike proteins actually can be detected even after 4 months of the second dose of the vaccination. So that's a very, very interesting thing, although I didn't think that it received enough traction. Now, there are some links over here. This is the exosome. So my question was, when I read the study, that can an exosome enter brain, or could it create cross the blood-brain barrier? Because there are inflammations in the brain as well for COVID, or sometimes in the vaccine as well. So could this be a reason? So the authors did not discuss this. This is a separate study in the NIH library, which shows transport of extracellular vesicles, these are the exosomes, across the blood-brain barrier, brain pharmacokinetics and effect of inflammation. So we'll look into that as well. Then this is another study here. And then what is tumor necrosis factor? This is the spike protein by itself can be harmful to the cells. And how does that work? And this is the immune system and its mechanisms, which we have discussed many times. So these are the links present in the description. Let's now go to my favorite way of studying these things. So this is the studies PDF. And let's go to my drawings. So gifts for humanity are continuing. This is the exosome. So what would stay in your mind for the time being are a few questions. Number one, what are exosomes? Number two, where are they formed? Number three, how long they're formed? And number two, why are they containing the spike protein on them? Number five, what happens to this spike protein? What does it do? So all of those we would discuss. So exosome with spike protein in the circulation, in the blood circulation. So here, this is an exosome. This is my depiction of the exosome. Exosomes are actually built inside our cell. And down in the lecture, I would explain a little more. But for the time being, when a cell uses a lipid nanoparticle and messenger RNA arrives in it, that cell will make spike proteins. Those spike proteins would then enter another vesicle called endosome. Endosome's function is to kind of break them down. That endosome can then package up these spike proteins, and that packaged material is called exosome. Why? Because it can exit the cell. So it actually gets out of the cell, containing those pieces of spike protein or the whole spike proteins with it. And then that exosome, it is small, it is lesser than 200 nanometer in size. Remember that SARS-CoV-2 itself is anywhere from 80 to 150 nanometer in size. So that exosome, that little particle formed within our cell, is small, almost similar or very small as well, like a virus. So it enters the blood vessels. The question then becomes, how did it enter the blood vessel? Did it enter the blood vessel because there was inflammation at the site of the um, injection, and that inflammation opens up the blood vessels, and through those opening, the exosomes are entering the blood, or they are just able to cross through the endothelial cells and entering the blood, or they're going through the lymphatic route. That means from the site of injection, the exosomes enter the lymph, and from there the lymph node, and from there finally into the heart, and then the blood circulation. So that means there can be many routes. So why do we care? From the researchers' point of view, we should care because they think this is the mechanism with which the messenger RNA vaccines produce antibodies. They produce exosomes. Exosomes have spikes proteins on them. This is very similar to a Novavax vaccine structure, a particle attached with the spikes on that. And then those exosomes enter blood circulation, and then our immune system detect them and respond to them, and that is how the immunogenicity occurs. So they call it a novel mechanism, a new mechanism. This mechanism was not observed before. So this is their observation. And they also say that these exosomes that have these little spike protein, like little horns on those exosomes, they are circulating in the blood. They will go to the spleen, and they tested this on mice and proved it in the mice's spleen. They go to the spleen. Inside the spleen of the mice, those exosomes was picked up by the macrophages, or innate immune system cells, and then they activated the immune system over there. So the second part of the mechanism was that these exosomes were responsible for immune activation, especially cellular immune activation, that is CD8 positive cell or cytotoxic T cells, and their activation in the spleen. Now, that is an interesting situation. If this gets into the spleen and the macrophages of the spleen, whose job is to actually clear out such things, can these exosomes enter other tissues as well and cause inflammation there? In my opinion, yes, but authors did not say it. Then, can this cross blood-brain barrier? So again, authors did not talk about it. I just showed you a study. I'll discuss that a little more. Then, is the spike protein pro-inflammatory, which we know that it is. So having spike protein entering the blood and going into the circulation and the tissues, could that be causing inflammation in various parts? Then spike can disrupt cellular functions, we know already. And finally, zombie particles. These particles probably can be called zombie. There was a study, I have forgotten which study, was it Chinese study or from where. In the study, the researchers had said that we are observing zombie particles with the spike proteins on them in the circulation. And they said these are not viable viruses because they don't have the virus messenger RNA, but they still are viral dead bodies. So I think that what they were seeing was exosomes. They were not seeing viral dead bodies. So many of the previous studies can actually take advantage of this finding. Now, I want to be fair to the authors and talk about their summary, the way they presented it, and then we'll continue. So they said, to determine the mechanism of the messenger RNA vaccine, especially Pfizer-BioNTech, we analyzed the kinetics of induction of circulating exosome with SARS-CoV-2 spike protein and antibody following vaccination of healthy individuals. They said we wanted to see, now exosome is also called extracellular vesicle. I was talking with my son this morning and giving him an example. I said, imagine a car is a cell. And if you have something in the car, for example, let's say a piece of some debris or trash, and you hold that and bring that hand out, if you can look at my hand, outside the car, that is like presenting the antigen to show it to other immune cells. And the car being a cell. But if you are in the car and you have some trash, and if you just throw it out, that is an exosome. An exosome is a packaged particle that is exited from the cell. So here they said we wanted to see the exosomes. Will they be formed from the vaccine? Will they then come out of the cell? And they found it that yes. And then they also wanted to see that when the exosomes will be formed, will they participate in producing immune response? And they also saw that yes, whenever the exosomes went up as a result of a vaccine dose, the antibodies went up as well, especially after the second dose, 14 days after. They saw that the antibodies did not form after the first dose, but exosomes formed. And after second dose, exosomes and antibodies formed. And they continued on for 4 months. Okay, then they said results demonstrate induction of circulating exosome expressing spike protein on day 14 after vaccination, expressing spike protein on day 14 after vaccination, followed by antibodies 14 days after the second dose. Exosomes with the spike protein, antibodies to SARS-CoV-2 spike, and T cells secreting interferon gamma and tumor necrosis factor alpha increased following the booster dose. And I believe over here, when they're talking about booster, they are actually talking about the second dose. They have written a little confusingly in their document couple of things. At one point in the document, they say that exosome has S2 part of the spike protein. In the other parts, as you can see over here as well, they simply say spike protein on the exosome. So see here, exosome with spike protein. But then later, just one sentence, they also say exosome had S2 spike protein. This actually also tells me why monocytes in the brain may have S1 in them. If there are exosomes with the spike protein and they entered the brain, if they entered the brain, and the monocytes then pick them up, that is Dr. Bruce Patterson's theory or the study that S1 is present in the monocyte. This may be the vehicle through which the S1 is reaching those cells. Again, I cannot connect these two. Now, then, transmission electron microscopy of exosome also demonstrated spike protein antigens on their surface. So not only there were exosomes, they had spikes on them. Exosome with spike protein and antibodies decreased in parallel after 4 months. That's a long time to have exosomes being so exosomes are produced all the time. They are extracellular vesicles. They are being produced and released from the cells all the time, not a problem. But exosome with spike protein after vaccination up to 4 months still coming out, although significantly reduced. These results demonstrate an important role of circulating exosomes with spike protein for effective immunization following mRNA-based vaccinations. This is further demonstrated by their test on the mice. So that is the basic summary. Now let's go a little more in the detail. What did they do? They had 8 patients. There was some control, about, I think, 50 control, but these were the 8 patients who were volunteers who said, we are ready to give you blood before the vaccination, on the day of vaccination, 7 days after, 14 days after the first dose, then 14 days after the second dose, and then 4 months after the second dose. And this research was done in St. Joseph Hospital, Phoenix, Arizona, and they partnered with the pathology department of Yale University. So not a tiny study, or it's a small study, but the heavyweights were working on it. So what did they do? They collected the plasma or the blood from individuals. So let's say this was a dose 1. So they collected the blood before dose 1, then on the day of dose 1 as well, then 7 days after, 14 days after the dose 1, then 14 days after the dose 2. This is dose 2, and then this is 14 days after, and then 4 months after the dose 2. This is what they had taken. And what they did was they isolated the exosome from these people's plasma. And they said that our exosome were according to the definition or accepted definition of exosome, and that was lesser than 200 nanometers. Then they did, of course, an electron microscope doesn't look like this, it's just my cartoon. Then they used transmission electron microscope to then look at these exosomes. So do you want to see those exosomes? It is, you know, that these things make me so excited to look at. So let me show you. This is an exosome in the blood circulation with the spike proteins on it. These arrows are showing spike protein. This is a 50,000 times increased or zoomed in view. This is SARS-CoV-2 spike protein with exosome. This is control. This is vaccinated individual. This is no conspiracy theory here. They are actually saying that this is a new mechanism with which messenger RNA vaccines work. But there is a proof that vaccine generated spike proteins are loaded on exosomes and then they are released, which then they go in the blood, and of course then they are picked up in various tissues and then they are responded to by our immune system. In this case, they used those exosomes, brought them into mice, and they saw that the spleen of the mice had the immune cell, and all of our spleens have immune cells. They responded. So anyways, going back here. Then what they did was they did the antibody visualization as well. They wanted to make sure that if exosome is present in the blood, are we going to see antibodies as well? Are we going to see immune system responding as well? And they saw that after the first dose, 14 days after, exosomes with spike protein were present, antibodies were not. Then 14 days after the second dose, exosomes were present and antibodies were present, and they stayed high for 4 months, and then they started waning, both exosome and antibodies. Now they used controls, and the controls were positive control who would also have these exosomes. Infected individuals, there were 10 COVID-19 infected. 2-dose vaccinated already, 20. And then healthy individuals, no infection, no vaccination, 20, so that they can compare their plasmas. Then what they did was, this I think was very clever, although sad for the mice involved. So what they did was they took those exosomes. So imagine if I get vaccinated 14 days after the second dose, they take my plasma. From that plasma, they extract those exosomes that has spike proteins on them, and then they inject those into a mouse. This is like a vaccine to the mouse, just like we can get Novavax. So now the mouse developed antibodies against SARS-CoV-2 because spike protein was sticking on top of these exosomes. This proved it. So they gave these little exosomes to this poor mouse, thanks to the mouse for the contribution, subcutaneous. And then they started taking mouse's data, blood samples, on day 1, 7, and 21. Sorry, they were giving it 100 microgram of these exosomes on day 1, 7, and 21. And this didn't have any adjuvant. That means there was nothing to trigger the mouse's immune system to make something. Just plain exosomes with these spike proteins. On day 30, the mouse was sacrificed. They took out the spleen and the plasma, and spleen had activated cytotoxic T cells, CD8 positive cells that were secreting, and the other T cells that were secreting interferon gamma and tumor necrosis factor alpha, which are pro-inflammatory markers and immune activators. And the plasma of the mouse was given exosome, but the plasma of the mouse showed antibodies that had developed against SARS-CoV-2. So they are saying that this is the mechanism in humans as well for how we develop antibodies against SARS-CoV-2 through this route. Now, what is the difference? What is the new thing here? We know that the normal expected mechanism, and I have discussed that like 200 times by now, is that here is a lipid nanoparticle from the vaccine, it enters a cell, muscle cell, fibroblast, whatever cell is present in that deltoid area where it is. Then this messenger RNA comes out to the lipid nanoparticle. It goes to the ribosome. Ribosome then makes spike proteins. Those spike proteins enter the endosome. We have done this discussion many times. The endosome is the shredder, and it shreds them and it breaks them. And here is the addition. In the case of messenger RNA vaccines, this endosome is also making extracellular vesicles, meaning small little pockets, lesser than 200 nanometer in size, lipid particles. So cell said, hey, you're going to send me a lipid particle, I'm going to make my own lipid particles. So it made a 200 nanometer size lipid particle or exosome, put the spike protein on it. Now, if I look at their paper, most of the time they say spike protein. In one sentence, they also say S2. So I'm going to say spike protein or the pieces of spike protein. They put that on it and they released it. Cell started releasing it. Now we got exosome in the blood with the spike on it. So then what happened? Then they speculate, they have a conjecture, and I think they're correct on this one. They say that these exosomes are then picked up by antigen presenting cells. Antigen presenting cells are immune cells, for example, macrophage, dendritic cell, and B cells. So antigen presenting cells, they pick up these exosomes and they digest them and they bring those spike proteins, and then they show those spike proteins to the CD4 positive cells, that is acquired arm. When they show it to the CD4, now there are two possibilities. Either we go B cell route or humoral immunity route. What happens is interleukin 4 is present, interleukin 12 is not present. That is a condition to go this route. And then the naive T cell becomes T helper 2. Then the T helper 2 cell releases multiple cytokines, including 4, 5, 6, and 10, which causes a B cell to proliferate and differentiate and become a plasma cell. A plasma cell is the one that is making antibodies. So this is how exosome is helping make antibodies. We're not talking about the virus, we're not talking about the spike protein. We're talking about exosome laden with the spike protein entering the macrophages or dendritic cell and then triggering the immunogenicity. What this means is immunogenicity is not only occurring in the deltoid, it is occurring wherever the exosome goes and it is picked up in the tissue, especially spleen. Why spleen? Because spleen's job is to pick up such complexes and clean them from the blood. How does it do that? Inside the spleen are macrophages and the other immune cells sitting there. They mop up such material, and not only they mop them up, they actually start the immune response as well. I would have wished if these researchers looked at the blood-brain barrier or other barriers as well to see how well the exosomes were crossing there. They have not done it. So any discussion of blood-brain barrier is outside the scope of their study. Okay. The other thing that they saw in the spleen of the mouse, when they entered this exosome, and they speculate that the MHC2 presented to the naive T cell, there were presence of interleukin 12 as well in the spleen, which caused naive T cell to become T helper 1 cell. T helper 1 cell released interleukin 2. Interleukin 2 activates more T helper 1 cells, plus it activates cytotoxic T cells. These are helper T cells or CTLs. These are CD8 positive cells. They have granzymes and perforins. They can make holes in the bad cells or infected cells and kill them. And they saw that in the spleen of the mouse, these T cell had and immune cells had become activated, and they were releasing tumor necrosis factor. They were releasing interferon gamma. Interferon gamma is a very important factor, because what happens is innate arm activates the acquired arm by releasing interleukin 12 and interleukin 4 from someplace. In turn, the acquired arm amplifies the innate arm, if it is this pathway, that is cytotoxic pathway, by releasing interferon gamma. This is called interferon gamma IL-12 axis. They amplify each other. And the researchers found a lot of interferon gamma. That means immune system is activated. So researchers said, yay, immune system is activated. That also means inflammation occurred. That also means whichever tissue picked up this thing and did this, that would have inflammation. So then they also talked about the kinetics, meaning when did the exosomes form? What were the levels? When did the antibodies form? What were the levels of the antibodies? And so on. Now, they also tried nucleocapsid protein as well, in addition to the spike protein, and they saw that the nucleocapsid protein did not do this. It was the spike proteins that did it, or messenger RNA vaccine did it. So here, what they saw was, after the dose 1, 14 days after, exosomes with S2 or spike protein. So they used, in this paragraph, they used the word S2. So I just have to be loyal to their paragraph that they said S2 part of the spike protein. And after and they said antibodies were not seen at this time. Then dose 2, 14 days later, they saw a significant increase in the exosomes with spike. They saw an increase in antibodies. And then they said 4 months later, the exosome with spike were less. Now, exosomes are always there, but exosomes containing spike were less, and the antibodies were less. So they both waned together. For me, what was surprising was that they saw spike protein laden exosomes still less, but they saw them even after 4 months. This could tell us why some folks continue to have issues. And this could also tell us that maybe this is how the messenger RNA vaccines would produce support. And then as the exosomes are reduced and the antibodies are gone as well. So this is not the most durable mechanism, as we're seeing that the waning is occurring very often. But this is a novel new mechanism. Now, different study. This study is done. We've seen the mechanism. We have seen how it works. We have seen the summary. Now, a different study. This study. Transport of extracellular vesicles, or exosomes, across the blood-brain barrier, brain pharmacokinetics and effects of inflammation. This is published 2020. And I'm just going to use a little bit of the mechanisms from here. So what they're saying is, here is a blood-brain barrier. This is a blood vessel which has entered the brain tissue. These blood vessels are really guarded, that they should not bring trash and junk in the brain, because a brain cannot handle it, and we only have one brain, and then it is closed in this the skull vault, and if brain swells up in there, we can have severe brain damage. So our brain does every possible way to make sure that the things do not just enter it willy-nilly. So here is the blood vessel that entered. Brain blood vessels have a specific structure, that their endothelial cells, or their cells that are making up the vessels, are very tightly, closely knit. So they have less spaces between them to allow things to get out. That's one. Second, they have another wrapping on them, which is called pericyte. Imagine the cross-Atlantic gas pipeline, and there is water around this, and you have to wrap it in multiple layers to protect it. So similarly, pericytes is another wrapping of cells around this to further protect. And then there is a third layer around that, which is called astrocyte layer. And these are the pseudopods, or the pods of the astrocytes. So 3 layers are protective. Not only they are just simple protection, but they are also very wise cells. And what they do is they decide what thing can be brought into the brain, or what thing would be allowed to get out. So they have a lot of transporters, a lot of adsorption mechanisms to decide and create a healthy nutritional balance for the brain. Exosomes can cross all of this. And here is that study. So if I can just read this, in summary, this is the last part I'm reading. In summary, all exosomes tested, so they tested about 10 types of exosomes, here readily cross the blood-brain barrier, but at varying rates and by a variety of vesicular mediated mechanisms involving specific transporters, adsorptive transcytosis, and a brain to blood efflux system. They are talking about various systems that are used to allow the vesicles to come in or go out. Now, there is no study that shows these exosomes to enter the blood-brain barrier, but exosomes generally can do that. So this is another study. So of course, this is a microglia. What is a microglia? Microglia is the macrophage of the brain. Brain has its own immune system. It does not want to work with the body's immune system. It does not want to do anything to do with them. But it has its own immune system and it has its microglia. Microglia are the macrophages. They will work and pick up these exosomes and they would cause inflammation. And who is the scared party here? The nervous tissue. The inflammation here is going to cause the neuron to become damaged. Not only neuron, other tissue would get damaged as well, but neuron is the more important thing. So that is another interesting possible connection. The authors did not talk about it. Then, this is another study, and there are tons of study about how spike protein themselves also cause various kind of cellular signaling abnormalities and inflammation. We know that we talked about ACE1 and ACE2 imbalance through the spike protein. We talked about nuclear factor K beta activation. We talked about nitric oxide depletion and so on. So there are possible mechanisms. For example, on the macrophages, innate immune arm cell, when the spike protein is detected by Toll-like receptors, that would cause nuclear factor K beta pathway to become active, which would cause inflammation. On the other cells, for example, blood vessel cells or heart cells or other tissue cells, ACE2, when that connects with the spike protein, other than the function of the ACE2, there is an abnormality in nitric oxide production, which is very important for reactive oxygen species defeat, plus the vessel relaxation. So the hemodynamics and endothelial systems health is affected. The blood vascular system's health is affected. That would also mean clotting could occur. And we already know ACE1 and 2 imbalance. So this is a study about that. Then here is another study that talks about the spike protein of SARS-CoV-2. This is the Journal of Biological Science. Chemistry. The spike protein of SARS-CoV-2 induces endothelial inflammation through integrin A5 beta 1 and NFK, nuclear factor K beta signaling. So that is also a possibility. So this is a discussion. This is just the mechanism. So this is the discussion for today. I hope that this made sense. Like last time, is there a need to do a separate 10-minute summary without any diagrams, or this makes sense? Let me know in the comments and we'll go from there. Now, for the rest of all of us, we're going to see each other on the chit chat in a few minutes. Thank you very much. Stay safe and happy and healthy. There is a link in the description if you would like to buy the premium content from Dr. Bean, and for the price of 2 lunches, and that's one-time price, not even recurring, then take advantage of that. And in addition to that, if you would like to support this work, there is a link to buy me a coffee or use PayPal or become patron, whichever you like. And even if you didn't, that is fine as well. Your liking, subscribing, and sharing helps too. So bye for now.